But how? The molecules made in these structures are messengers sent to other regions of the brain. And Dr. Young found that what was different between the voles was how able they were to receive the chemical messages. The brain regions receiving these messages are involved in feeling that something is pleasurable or important. The prairie voles are quite attached to their partners, and in learning of social behavior, allowing them to cooperate in long-lasting relationships. More recently, Dr. Young has been exploring applications of what is going on in voles to what is going on in us. Although some of the complexities in our social relationships are an integral part of being human, we do have much in common with other animals, at levels ranging from the molecular to the behavioral. Moreover, what we are learning about these commonalities with other animals may help us to understand differences among humans, including differences between those of us who are sometimes called neurotypical and those of us on the autism spectrum. Why do we interpret and respond to social cues in different ways? What is different about what messages are sent through our brains and how they are sent and received? Some of the answers to our questions may soon be coming from scientists like Dr. Young and from the voles. An author of several dozen publications in journals such as Science, Nature, and Neuron, Dr. Young is the William P. Timmie Professor of Psychiatry and Behavioral Neurosciences at Emory University and Chief of the Division of Behavioral Neuroscience and Psychiatric Disorders at the Yerkes National Primate Research Center. Last year, he established and now directs the Center for Translational Social Neuroscience. He has been an accessible, fascinating communicator in venues ranging from the annual Society for Neuroscience Conference to Dateline NBC. It is my great pleasure to introduce Dr. Larry Young.
Okay, good morning, everyone. Good morning. And thank you for everyone coming here. And I want to thank Chuck Niederreiter and all the rest of the organizing committee for inviting me to participate in this conference. This really is truly an honor for me. So I'm going to talk about the work that we've been doing over the last, say, 15, 16 years, trying to understand the neurobiology of the social brain, the neurobiology of social relationships. And this picture, I think, nicely illustrates some of the relationships that we experience in our lives. As humans, we all know how important those relationships are. And there are several different kinds of relationships that I will talk about today. And some aspects of these relationships we may consider to be uniquely human. But I also want to argue that those relationships, the biology behind those relationships, are evolutionarily very ancient. And we can see traces of these in other species. And some of those, for example, the relationship between the mother and the offspring. This is not uniquely human, of course. We see this in all mammalian species. In all mammals, there's a strong bond that occurs between the mother and the child. But in humans, we also have different relationships. For example, the relationship between the father and the child. The father is often present in most societies. But we also have a relationship that forms between the mother and the father. This seems very natural to us, but if you look in the animal kingdom, around 97 to 98% of mammals, there is no relationship at all that exists beyond the sex. Once the female becomes pregnant, she's off on her own to have the babies and raise the babies on her own. The male's interested only in finding another female to produce more offspring. So I'm interested in the biology behind that. Why are we different? And if you want to understand the biology, you have to have some means of interrogating the system. And in humans, a lot of times we can learn something by studying humans, for example, with brain imaging. fMRI allows us to peer into the brain to see what brain areas are activated during certain processes. But actually, my degree was in biochemistry. I'm interested in the biochemistry underlying behavior and the genetics underlying behavior. And those things are very difficult to understand using human subjects. And so I turned to an animal model, as you heard a few minutes ago. And I turned to these animals here. These are prairie voles. Okay, these are little hamster-sized rodents from the Midwestern United States. And unlike most of the other animals that are studied in laboratories, mice, rats, most other things that you can think of, these guys are socially monogamous. And I want to make the point early on, they're socially monogamous, which means that once they form a partnership, they usually stick together for life. But if the male is out wandering along in the prairie and a female in estrus comes by, a female in heat, he may actually mate with that other female. But the important thing is that he comes back home to his partner at night. So what we are studying, what my entire talk here is not about sexual exclusivity or genetic monogamy. What I'm talking about is the formation of relationships between these two, the formation of an attachment and a bond, okay? By the way, the female may also do the same thing. So I just want to say a little bit about the evolution of monogamy. I think it's very interesting that it occurs only in 3% of species, but it occurs in 3% of species that are often unrelated. So it arises independently multiple times. And I'm also interested in how do we get new behaviors from the ones that previously existed. Now, I just want to say something about why does monogamy ever evolve in the first place? When is monogamy adaptive? If 97% of species are not monogamous, then it seems like in most cases it's more adaptive for males, for example, to try to mate with as many females as possible. And there are two examples that I'd just like to highlight of when it makes sense for an animal to form these bonds and cooperate. And one is in the case when it takes two to raise the family. And that may be the case in human evolutionary history as well. But imagine that resources are hard to come by, or there are predators in the environment. If a male, his brain is constructed in such a way to not form a relationship with his female partner, and he is completely promiscuous, he doesn't help raise those offspring. He may have, in the case of voles, hundreds of babies, but none of them may be able to survive, and so none of his genes get passed on to the next generation. Whereas the male who works with the female, protects the nest against weasels, may actually pass his genes to the next generation. Another example is sometimes it may just be hard to find a good partner. So when you find a good partner, stick with that partner. Okay, don't risk it. And a good example of that is the deep-sea anglerfish. These guys live deep in the ocean, dark ocean. They have this little antenna that's bioluminescent here. And you can imagine that a male may spend half of his lifetime just trying to find a female to mate with. So when he does find one, he should bond with that partner and form a long-lasting attachment. And that's exactly what he does. In this case, these animals, the male finds the female and he bites her. Bites a hold, doesn't let go, his blood vessels fuse with hers, and he basically disintegrates until there's nothing much left but his body's most important parts, and that's his hypothalamus and his gonads. And you may think he has lost a lot of independence and quality of life, but the important thing is the genes that cause his brain to form in such a way to engage in this behavior will be passed on every time she is ready to spawn because she is right there. Whereas the other males in the species who decided not to engage in this behavior may not pass their genes on. So that's how natural selection works to create new behaviors. Now, I mentioned I'm interested in the evolution of behavior, and I just want to make the argument. I believe that monogamy, in some cases, the ability to form attachments, evolved from this evolutionary ancient behavior of the mother-infant bond. Brain systems that are involved in that very powerful bond have been somehow tweaked to give rise to this new social attachment. And I want to tell you why I believe that, and I want to introduce a molecule that plays a critical role in social relationships. This molecule is called oxytocin. It's been known for a long time to be involved in two processes critical for reproduction. One is uterus contraction during labor, and in fact, many physicians now can schedule a delivery just by injecting oxytocin into a pregnant woman. After the baby is born, oxytocin continues to be secreted, and acts at the breast to stimulate milk ejection. So it's responsible for giving birth and also providing nutrients to the baby. Well, just a little bit to show you about the neurobiology of that. Oxytocin is produced in neurons up in the hypothalamus, which we heard about in the introduction. And in most cases, it is secreted into the bloodstream where it acts on the peripheral tissues. Stimulation of those tissues, for example, vaginal-cervical stretching during birth causes more oxytocin to be released, or when the baby suckles, stimulation of the breast causes more oxytocin to be released. So there's a positive feedback. But we now know that oxytocin not only is responsible for the physiology of reproduction and birth, but it acts in the brain to transform the mother's behavior. So we know this, for example, from studies in rats. These were done in the early 1980s. Now, you may not realize this, but virgin female rats, like most species, they don't like babies. Okay, they find pups very annoying. It's very much like, I think of it as when I fly across the country and there's a baby a few seats away on the airplane and they're crying the whole time. I find that to be very annoying. But something happens when the female goes through pregnancy, so that just within hours before she gives birth, a transformation occurs in her brain, and now pups become very attractive. And she will even cross an electrified grid to get access to those pups. And we know that part of that transformation is due to oxytocin, because you can take a virgin female rat and inject oxytocin into her brain, and suddenly she will be attracted to pups. Similarly, in sheep, so rats will basically take care of any pups you put in the cage. They don't bond with their own babies necessarily. They bond with their nest. You put other babies in there, they'll take care of them. Sheep are different. They give birth in a breeding season. Lots of females are giving birth at the same time. When the lamb is born, it's able to walk around. So there's lots of lambs walking around. So the ewe has to not only say, hey, I like babies, but they have to bond with their particular baby and say, I like my baby. And I'll butt away at the other babies. And we know that oxytocin is responsible for that two ways. One, as I mentioned, vaginal-cervical stimulation stimulates oxytocin release. Well, farmers have known for a long time that you can induce a female sheep to bond with and adopt a lamb through vaginal-cervical stimulation. Or if you inject oxytocin directly into the brain, the female will bond with the male. With the baby. So that's all I'm going to say about maternal behavior now, because I wanted to switch to this other aspect of relationship, and that is the bond between two adults. And this is a behavioral test that we use to test that. It's called the partner preference test. And so this is how we ask whether two voles are bonded to each other. We put the animals together, and during this time they can mate, or we can prevent them from mating, or we can give them various drugs to interrogate the neurochemistry underlying the process. Then we can separate them, and then we give them this choice. We take the partner, in this case we're taking the male partner and putting a collar around his neck so that he is restricted to this cage, freely moving in this cage but can't get out. We take a novel male, a stranger, put him in the third cage over here, and then we drop the female in the middle and just ask her, who does she like to spend her time with? And if she's bonded with that male, she'll spend time with her partner. If she's not, she'll spend equal time. And we know that mating facilitates this pair bonding process in voles, but it's not absolutely essential. And I just want to show you a video, if it works. This is actually, in this case, we've tested, this is the male in the middle, and we put this female partner over here and a novel female over here. Now, keep in mind, in most species, if a male is mated with one female and he sees a different female, he actually prefers the novel one. But you see, he showed a little bit of aggression towards this novel female that wasn't his partner. Now he's going to come over to see his partner that he spent the night with the night before, and you can see he behaves very differently to her. So we can do large-scale experiments with 30, 40 animals at a time. This behavior is all quantified by computer analysis, so we can really tell whether they have formed that attachment. And because of oxytocin's role in the formation of the bond between the mother and the baby, Sue Carter and Tom Insel, a couple years before I got involved, wanted to ask whether oxytocin might be responsible for the bond between the female and the male. And in fact, they found that if they took a female vole, put her with a male for only 6 hours, a very short amount of time, they were not able to mate. They did not form this partner preference. But if they infused oxytocin into her brain just immediately before placing her with the male and then testing her, she formed that bond. So this shows us that this one molecule plays a critical role in the formation of the attachment between these two individuals. Now, one of the really cool things I think about this system is that there are different kinds of voles, different species of voles that look very similar, but their behavior is very different. So, for example, the prairie voles, as I mentioned, not only are they socially monogamous, but they're very highly social. They crave social contact. They spend much of their time interacting with each other, grooming each other. If you contrast that to the meadow voles, these animals are pretty much solitary. They don't have the same brain neurochemistry that makes them want to interact. They're promiscuous. They don't form bonds at all. Males don't form attachments at all. So we can ask, what is different between these guys that can form these attachments versus those that cannot? Why can't the meadow vole form this kind of bond? Well, you might think they have different levels of oxytocin, since oxytocin promotes the bonding, but that's not the case. Oxytocin is exactly the same. What's different is the receptors. So this illustrates basically how neurons in the brain communicate. Neurons release neurotransmitters or hormones. This is one side of the equation, and they act by acting on receptors. So you need both of these to have this communication. We found that there's no difference in the oxytocin, but if you look at the receptors, there's quite a big difference. Now, how do you see receptors in the brain of an animal? This slide is just to illustrate that. You can take the hormone and put a radioactive molecule on that hormone, incubate it on the brain slice, and cells that have the receptors will bind to that hormone. And then you have a radioactive spot on the brain. And you expose this to film, and you get brain images that show you where the receptors are concentrated. And if you compare the prairie vole to the meadow vole, what you see is very different locations of receptors. That means different parts of the brain respond to the oxytocin when it's released. This area is really important. I wanted to highlight this. This is an area called the nucleus accumbens. This is an area that's involved in reward, reinforcement, and addiction. Cocaine acts in this area to produce addiction, and prairie voles have high densities of oxytocin receptors there, whereas the non-monogamous species don't. That's pretty cool. It's actually easier to tell this species apart, as far as I'm concerned, by looking at their chemistry in the brain than it is by looking at their physical appearance. I think that tells you something about the power of the biology behind this. And we know that these particular receptors are important for bonding because if we inject into this area of the female vole a blocker to block those oxytocin receptors, the female, even if she mates with a male for 24 hours, does not form a bond. Okay, so these are critical. We can actually do things in these animals that we could never do in humans, so we can put little probes into the brain that can collect molecules as they're released. And we can see that if we put this into this area of the female vole, when she's sitting alone, there's no oxytocin being released. But if she's placed with a male so that they're able to mate, the vaginal cervical stimulation that occurs during the mating process causes a dumping of the oxytocin into the brain. Now, oxytocin may play an important role in male bonding as well, but most of the work has focused on a different molecule. It's almost identical to oxytocin, but it's called vasopressin. And one of the cool things about vasopressin is that it's one of the more sexually dimorphic molecules in the brain. What does that mean? It means that males have much more. And this is just a little section through a part of the brain. The white spots are the vasopressin. You can see that males have much more than females. And it's responsible for male typical behaviors in a lot of different species. For example, territorial behavior. Animals like to scent mark. Like if you walk your dog, the dog urinates all over the place to scent mark his territory, right? Hamsters do the same thing, and if you inject vasopressin into their brain, they immediately scent mark their territory. It's also involved in aggression. But in these voles, it does something on top of that. It's responsible for the male bonding with the female partner. And we know that because we can infuse the vasopressin into a male, put him with a female, even if the female doesn't want to have anything to do with him. After 6 hours, he's bonded with that female. So I think this has some interesting implications for differences between the bonds between the female and the male and the male and the female. It seems that the female bonding evolved from the neurochemistry and the neural circuits that are involved in the maternal nurturing. So the female to male bond is a very nurturing bond, whereas the male to female bond may evolve from tweaking of the systems involved in territorial behavior. The female is really part of his territory. And I'm only speaking about voles here. I'm not really talking about humans yet. I will get to humans in just a second. I just want to make some important points about voles. Again, if you look at the vasopressin system, there's no differences in where the hormone vasopressin is located in the brain, but the difference is really in the location of the area that can respond to vasopressin. So here, prairie voles have high densities of receptors in this ventral pallidum, which is connected to the nucleus accumbens that I told you about earlier. That's also involved in reward and addiction. And we know, so in the metavoles have very few receptors there, and we know that those are critical for bonding because if we block those receptors, and we can inject the oxytocin vasopressin receptor blocker into different areas. If we block it in this area, the male prairie vole does not form a monogamous bond with the female. So I just want to talk a little bit about, how is it that these molecules can act in the brain to form a bond? Because that seems like a very complicated thing. How do you go from a molecule to a bond or to a preference? So I just want to highlight my vision of how this works. I told you oxytocin is involved in maternal behavior and maternal bonding, but we know from studying mice that there's a more subtle effect, or actually I think it's a really more important effect of oxytocin, and that is that it's responsible for the ability to process social cues of others. So all of you out there, there's thousands of people here, all of you have heads that are about the same size, eyes that are about the same distance apart. Nose, you have a nose here, you have a mouth. But my brain is fine-tuned to look at each and every one of you and pick out the individual characteristics that make you unique. Okay, that's kind of a social recognition. Mice have the same ability, except they do it by smell. As you can imagine, all mice look pretty much the same, but they all smell different. And mice use oxytocin and vasopressin receptor systems to amplify that social signal so that they can tell each other apart. How do we know that? Mice that don't have oxytocin have social amnesia. They can't recognize another mouse. So this is a very simple experiment. We take a mouse. The white mouse is the experimental mouse. This is another mouse that we put in the cage. When they meet, they greet each other by sniffing each other. We take them out and we put them back in a second time, and we quantify how long they sniff each other. We do it again and again. And each time they meet each other, they sniff each other for less because they know each other. Then the last time we put in a new mouse. They don't know each other, so they sniff each other for longer. If we do this with wild-type animals, you see the nice decline. If you do it with if we do this with wild types, you see the nice decline. If you do it in an animal who we've genetically engineered not to have any oxytocin, they can't remember 5 minutes later. They can remember other smells, lemon scent, almond scents, but they can't remember the smells that are used to tell each other apart. So from this work, we know that oxytocin and vasopressin make social signals more salient. What we also know is that in voles, the pair bonding process requires dopamine, which is involved in reward, reinforcement. It's why cocaine feels good, or food, or other pleasurable things. Other systems, opiates, which is in morphine, involved in making pleasure, is also required for the pair bond. So putting this all together, here's what I think what's happening in a brain when the animal is forming a bond. This animal is mating. Imagine the animal, this is a male rat, for example, he mates, activates an area called the VTA. That's where dopamine is made. Dopamine is then released into this nucleus accumbens and ventral pallidum, the reward, reinforcement area. And that's what makes sex rewarding. And we know that even for a rat, sex is rewarding because a male rat will press a lever many, many times to get a female rat to drop out of the ceiling. But when when a prairie vole mates, so when a rat mates, they smell, this is a female, she's in estrus. That was really good. I'm going to try to do that more and find more estrus females when a prairie vole does it. Because these receptors that are involved in social discrimination are concentrated in the reward area where the dopamine is being released at the same time, you get a linking of the encoding of the social cues of the partner with the reward of the social interaction. So then they want to spend time with that individual partner. So it all has to do with the fine-tuning of the perception of the social cues. And so they recognize who was that I was with when that was pleasurable. I'm going to skip this part. So that hypothesis, that model, suggests that the reason that prairie voles can bond and meadow voles cannot form these relationships is because the prairie voles have the receptors there, allowing them to link the social encoding with the reward. The meadow voles don't. We tested that in the laboratory by manipulating gene expression. We took the vasopressin receptor gene from the prairie vole, put it into a virus, injected that virus into the brain of a meadow vole. Only the gene, only this one single gene, and put it into a meadow vole to create a meadow vole whose expression of this one gene looked like a prairie vole in this one brain area. And then we placed them with females. And we found that this species, which never should form a social attachment, now because we've changed this gene expression profile, is able to form a social attachment. So this shows you that evolution of behavior can occur simply by changing the distribution of a single gene, acting in particular brain areas, because those circuits are there already present to subserve other purposes like reward and reinforcement. Now, I'm interested in diversity of behavior. If you look in different species have different behaviors. Behavior evolves very rapidly. And I just show you this because I think that this is one of the coolest aspects of this oxytocin and vasopressin system, is that there is such diversity in receptor expression patterns. Different species express the receptors in very different ways, and I think that this is what gives rise to differences in social behavior. And it even relates to individual variation in behavior. And if you remember, in the beginning I told you that virgin rats don't like pups. Well, when you look at prairie voles, they're different. And about 60% of virgin female prairie voles, they are spontaneously maternal. They like pups from day one. The 40% do not. If you look in the brain, that difference in behavior is related to differences in oxytocin receptor. These are the same species. Now we're talking about individual variation. This is important for humans because we are all the same species. Here we're talking about individual variation. There are differences in receptor densities between individuals, and that predicts how they will respond to babies. So we can begin to understand the biology of individual variation. I want to say a little bit about how does this diversity evolve? How does it come about? Can we track it down to DNA? So here's a question that I began looking at many years ago now, is the difference between the two species in behavior is related to differences in expression of this gene. How do you get differences in gene expression? Can you go down to the DNA level? Now let me tell you a little bit about genes. This is the gene. It has a coding region that makes the protein that does the job, like the receptor, but it has a set of instructions in front of the gene that determines whether this protein is being made or not, or in what part of the brain it's being made, okay? When we looked at the genes between these monogamous and non-monogamous species to see what was different, the red part here is the part that makes the protein, which is identical between the two species. The genes in general were almost identical between the two species, except for in the part of the gene called the promoter, where there are instructions that determine how much of the gene is being expressed. The prairie vole had a repetitive DNA element that was much longer than the montane or meadow vole, okay? But the cool thing is that there was a lot of individual variation among prairie voles. So some prairie vole males had long versions and some had short. So this gives us an opportunity to see, is there a relationship between this DNA element and gene expression in the brain and behavior? So what we did was to basically determine how long this was in a bunch of our prairie voles, and we found prairie voles that had only long copies and put them together to make a male and a female that had a long microsatellite. We had some that had only short microsatellites, this little repetitive DNA element. So we created different lines of voles that differed only by this one little piece of DNA. And we put them together, let them have offspring, and then we tested their offspring's social behavior. And just to summarize very quickly, the length of this little piece of DNA predicted how much of this receptor was expressed in parts of the brain, but it also predicted their social behavior. Individual prairie voles with a short little, this microsatellite element were less likely to form an attachment with a partner than the ones with the long microsatellite. Now, I'm going to get back to this. I'm almost to the human part. But I want to say one more thing. So far, what I've suggested to you is that genes play an important role in the species differences and individual variation and behavior, but we also know that experience plays an important role. We did experiments where we took prairie voles and raised them by mother and father, a biparental group, so both parents were present, and we compared that to pups that were raised by the mother alone. So the mother had to go off and get food, sometimes left the pups alone. And we found that that also shaped the offspring's behavior. I know this slide is busy, but I'll just make the point that if they were raised by both parents, and both parents were on the nest, they were able to form social relationships more quickly than the animals that were raised by the single mother. So experience has an impact. And we know that this is a relationship, that there's a relationship between experience and human oxytocin system as well. This was work done by Christine Heim at Emory, where she took women who experienced early in life either neglect or abuse or not, and looked at oxytocin levels in the brain and found that the more cases of abuse and neglect of the woman experienced, the lower the levels of oxytocin in her CSF. So even in humans, experience can shape these systems that, at least in voles, affect behavior. Now, I'm going to get to the point that we're all here for. How can we use this information to profit mankind? That's actually not, that's a joke. I'm not selling that. But this is not a joke. This is a really important question, I think. As biologists, we spend a lot of time and money looking at these processes in these little animals that run around in the prairie. Does it have any relationship to humans? Can we learn anything about human? And I want to show you some examples of where there's some very exciting parallels based on what's been done in voles that's done in humans. I just finished telling you about this little microsatellite in the vasopressin receptor gene that predicts expression in the vole brain of the vasopressin receptor. It also predicts the likelihood that those animals will form the social bond. Well, it turns out humans, we share most of the same genes with animals. In the promoter of the vasopressin receptor gene, there is also a microsatellite that is different and linked between different people. There are 16 different lengths of that, and I'm sure that every one of those different lengths are represented in this room. A group in Israel took brain tissue to ask whether the length of the microsatellite, short or long, predicted the amount of gene that's expressed in the brain, in this case hippocampus of humans, post-mortem tissue, and indeed the length of the microsatellite predicted how much of this receptor was expressed. Another group at NIMH did studies looking at brain activation, classified people based on the length of this little microsatellite, and found that in the amygdala, a part of the brain that's important for emotion and also social communication, that the length of the microsatellite predicted how the amygdala would be activated when the person was doing a face processing task. So this little gene affects how the brain acts. And then this is one study that I think is most highly parallel to the voles. This is a study done by a Swedish group who looked at 1,000 individuals that were in live-in relationships, and they genotyped to see how long the little microsatellite was. And this time they looked at, they didn't just break it up into short and long, but they looked at all the different possible 16 lengths. And they found that it had no impact on women, just like it does not in female voles, but for the men, if the men were homozygous, which means they inherited this particular allele from both their mother and their father, they were twice as likely to report a crisis in their marriage in the last year. They were actually twice as likely just to be shacking up with their partner or not committing to the relationship by legal marriage. And also the partners of those individuals express more dissatisfaction with their relationship. So this is an intriguing example of where gene polymorphism that predicts vole behavior seems to perhaps also influence to some degree human behavior. Now, the work that's been done on oxytocin in animals has really exploded in human literature recently. There are papers coming out almost every day. Now, in humans, it's apparently possible to deliver oxytocin through intranasally. So if you sniff it, it can pass the blood-brain barrier and get into the brain. And there's been lots of studies where people have asked, well, if it does this in voles, what does it do in people? Well, one of the first studies that came out was a study that showed that oxytocin enhanced trust. And so people who were sniff oxytocin were more likely to trust another person in an economics game. And now this is a website that you can actually go to. This is not my product, by the way.
Imagine for a moment that everyone trusted you. You would sell more, love more, and accomplish more than you ever imagined. Scientists have discovered a hormone that increases people's trust in each other. It is the human hormone.
It's the human hormone oxytocin, it went on to say. So this website, actually, they tell you this product, if you take it and spray it on you before you go out on an important business deal or something, maybe the other person will trust you. Does that make sense to you? If you put it on yourself before an important business deal, who's going to trust who? So this is why you need to be educated and think rationally about things before you go off and buy it. But actually, this product does not actually work. I do not recommend this because, as I believe I mentioned that oxytocin doesn't pass the blood-brain barrier very efficiently, and the only way that you can get any into the brain at all is through this intranasal delivery. But this is an interesting example of how companies try to take advantage of the science that's been going on. This says here, if you read at the bottom, it says Liquid Trust Enhanced. This is the new version. They had to update it. Has been specifically designed to give a boost to the dating and relationship area of your life. This upgraded formula still contains the same great oxytocin, but now includes powerful pheromones. So they're saying spray it on you before you go out to a singles bar, and maybe you'll attract someone and also maybe get them to trust you. Like I said, this is not a product that I endorse, but I do want to now talk about some other studies that have really highlighted oxytocin's general role in the human brain. There's been 3 studies now that have shown that intranasal oxytocin actually increases the amount of time that a person spends looking into the eye region of another person. The eyes are where we get much of our social information from. This is actually very similar to what I talked about in the mice, where they meet each other and they sniff each other to extract social information. Humans look into the eyes. Not only does it increase the amount of time looking into the eyes, but it also increases something that's called mind reading in this paper, which is not really mind reading, but it's the ability to infer the emotions of other people by looking at even just portions of their face. So extracting social information again. Oxytocin, this paper came out in 2010, showed that oxytocin actually increases some aspects of empathy. So people who took the intranasal oxytocin and looked at images such as these were more likely to feel that same emotion as expressed in those images. Interestingly, one of the findings of this paper was that females show more empathy than males, but if the males were to do the intranasal oxytocin, it would raise their level of empathy back up towards the level of females. This is another example that I think is very interesting. They found that oxytocin increases socially reinforced learning. And what is that? Let me just explain this paradigm. people were given a task to learn how to do. At first, they don't know how to do it. Later, they can learn based on trials. If they get it right, in the social reinforcement group, they got an image of a face smiling at them. That was reinforcing. Oh, I got it right. If they got it wrong, they got this face of a person grimacing at them. So based on that feedback, they could learn how to do it. Well, half of the people were given saline, which is the control, so they were not given oxytocin. The ones in blue here were given oxytocin, and what they found that if people were given the intranasal oxytocin, they learned how to do this task faster if they got a reinforcement that was social. If they did the same experiment, but they didn't get a smile or a frown, but they got a green light or a red light, they did not improve. So this, again, points out to the fact that oxytocin is relatively specifically, impacting social cues, social information. This just summarizes some of the things that I've already said that oxytocin has been known to do. It also increases the memory of faces as familiar, which is very similar to the mice that I mentioned. And I think that all of these suggest that what oxytocin is doing is increasing the saliency of social stimuli so that our brain pays attention to the social information that is around us. We're attuned to the social information. We see people as different from trees and objects. They're special. And we can see minute differences in muscles in the face and understand what that means to the other person. Now, I think that has some important clinical implications potentially. So here's an important question is can this work in animals ultimately not only tell us something about social relationships in humans, but also be used clinically to correct disorders where there are problems in the social brain? I think you probably all know something about autism spectrum disorder. This is a disorder that's characterized by 3 symptom domains largely. One that we mostly think about is deficits in social reciprocity and social engagement. Individuals with autism tend not to look into the eyes. They don't reciprocate socially. There are also problems in communication, language development, and repetitive and restrictive behaviors. They like the sameness. Now, one of the challenges of developing drugs to treat autism is that there is not one autism. It turns out autism is a heterogeneous collection of disorders that all give rise to a similar phenotype, a similar behavior. But there are now dozens, if not hundreds, of genes that have been implicated in the cause of autism. They all somehow work by different mechanisms, but they somehow give rise to a common disorder, which means it's going to be difficult to find a drug to correct a particular gene. So what we've been thinking about in the lab is how can we go about maybe not correcting the fundamental gene that's wrong, but correcting the disorder, maybe enhancing social learning. And based on the work with the animals, we thought, well, you can break up social learning into different cognitive domains. One is social reward and reinforcement. There's social information processing, and we know some of the molecules that are involved in this because of the work in animals. We know that oxytocin is involved in social information processing. We know that opiates and dopamine are involved in social reward and reinforcement. And we know from work by many others that glutamate and dopamine are involved in learning and plasticity. And the idea here is that if you can enhance any one of these with a drug, you might increase social learning. And indeed, there's some evidence for that. There are 4 studies now, I believe, that have shown that in individuals with autism, oxytocin, intranasal oxytocin, can increase some aspect of social functioning, at least within a few minutes after delivery of the drug. But one of the limitations of the intranasal oxytocin is that we don't know how much of it is actually getting into the brain and how far it can travel into the brain. So we've been thinking of drug discovery ways of actually increasing the oxytocin system in the brain without actually giving the intranasal oxytocin to be more efficient. So how might we do that? Well, imagine if you knew something about the oxytocin neurons in the brain. and you knew that there were receptors on those neurons, not oxytocin receptors, but any kind of receptor for other, that there are drugs for, that you could give those drugs to the individual that would then cause these oxytocin neurons to dump oxytocin directly into the brain, you may have a very profound effect on the ability to pay attention to the social world around you. And indeed, there is one molecule that we found in the literature, that this was not discovered by us, but that if animals are giving this drug, you have a huge increase in oxytocin in the brain. This is a natural molecule, but the important thing is that it targets a receptor. There's a receptor called MC4 receptor, which is just one it's just one example of a receptor that's located in oxytocin neurons that you might be able to target with a drug. And indeed, we have tested this in our voles to see if you were to give a drug that hits this receptor, would it promote them in bonding? Would it promote their bonding? And we found that when we took voles, inject them with this drug just before putting them together for a very short amount of time, it induced the pair bond even much more strongly than we ever could do if we injected oxytocin directly into the brain, and it's because we believe it's dumping it directly into the areas where these receptors are. So this is potentially a way of profoundly affecting the oxytocin system. Importantly, this memory of the partner lasts weeks after the drug is gone, so the drug doesn't have to be there in order for that memory to be learned or to be retained. And I think that study in animals has critical importance to at least to how we think about when we may use this in treating autism. So right now, there is no biologically based drug treatment that targets the social domain in autism. But imagine if, the most effective treatment of the social deficits in autism is early intervention and behavioral therapies, where the child is exposed to a behavioral therapist, and they learn from social reinforcement in these behavioral paradigms. Imagine if you could somehow, in this child, open up their social brain so that they paid attention, they looked into the eyes of the therapist, they read the therapist's emotions better. Maybe they would improve the efficacy of these therapy sessions. So this is one of the sort of things that I'm very excited about, the evolution of my own research, is that I started out looking at this bonding in these voles, and it may be that that we can use these voles to identify drugs that could enhance social learning in a vole, which may then be very able to be developed into a drug that could enhance social learning in a clinical setting, that might enhance the effectiveness of behavioral therapies and ultimately lead to improved social functioning in autism. And that's something that we are very interested in pursuing. And this is sort of the rationale for the development of this Center for Translational Social Neuroscience that was mentioned earlier. The idea is you get basic scientists like myself, who has never worked with a child with autism, but who knows the biology and the genetics of it, put them together with people who work in a clinical setting, but who don't know much about the biology, bring them together to try to increase that transfer of information and to try to come up with novel strategies. And that's the end of my talk. I just want to acknowledge the fact that we get support from National Institute of Mental Health, National Science Foundation, and Autism Speaks. And this is my current lab. Much of the work was done by other people, but you can see that they're a very social bunch. Thank you.
Thank you, Professor Young, for that wonderful, illuminating talk. At this point, I'd like to ask the rest of our panelists to join us up front at the table. And while they gather, I'll remind our audience how we do the question and answer period here at Nobel. The ushers will be passing out cards on which you can write questions. They will collect those questions and bring them to the front, and they will be passed to the moderator and asked in turn. For those folks watching on the internet, there is a link to Google Moderator and a Twitter feed, and you can ask your questions using those. Those are on the web streaming website. Thank you.
Yeah, 5,000 is probably the people in this room. No, it's probably the overflow. The overflow is pretty big here. Yeah, it's the basketball.
Okay, folks. Okay, folks, we're going to try to get the question and answer period started. So if you could find a seat, and we'll open up the mics on the panel here.
I have my question. Does anybody up here want to get a ticket for reserved seating and lunch? 2 lunches in the banquet? I have an extra ticket. Oh, does anybody up there want to buy a ticket for reserved seating and lunches? I have an extra one.
It'll look really curious.
Not tired of sitting in the bleachers yet.
Okay, quiet now.
Yeah, I have an extra ticket.
Okay, we're going to start.
It's very funny that this is like, yeah, mine's like right on mine. Yeah, I'm gonna.
Okay, we'll begin by asking a fellow panelist if they want to make some comments or ask questions, and then we'll move to the questions from the audience and from the internet. Thank you.
You have one?
Oh, okay.
Go ahead.
Am I audible?
Yes.
Amazing. Okay. Well, first of all, I thought that was an awesome lecture. In fact, I'm now extremely anxious about meeting that standard you set. But sitting there, I was thinking, oh, yes, oxytocin, the more the better, and my daughter's 16. If she ever brings home a serious boyfriend, I'm going to try to get a DNA sample and find out what his vasopressin promoter, length is.
There's a company that will do that.
Yeah. I'll get their address from you afterwards. Now I'm questioning why do I assume that sort of more oxytocin and vasopressin action is better? Are there any reasons, either individually or for the community, to be thankful for those people with the short genes that aren't as inclined to attend to social cues?
Well, absolutely. I mean, people have often said that some of the greatest scientists were not the most social people. Albert Einstein was not the most social, so he didn't spend as much time reading the emotions of others and more time, doing his physics. So, I think variations in these systems creates diversity in social behavior and social relationships, and there are places for all of that variation. Maybe not all of the variation, but much of that variation in society. Just like, it's the same, I think, as in voles. Sometimes it's the environmental cues, the environment may be such that bonding is best. Other times, when there are different amounts of food or predators, animals who are not, socially monogamous may produce the most offspring. So it's like the environment. It's in the context of the environment of how social one should be. In humans, you can have the extremes of that, where you may have sociopaths who no one has really linked sociopath with lack of oxytocin, but it would be interesting to look at that to see. I think that there's a lot of room for diversity in behavior, and shouldn't all be the same. And then there are disorders like Williams syndrome. There was one study that has suggested that individuals with Williams syndrome, which, by the way, this is a disorder where the children are very talkative and they're very trusting. They tend to overly trust others, and it's pathological. And there's some suggestion that there may be high levels of oxytocin there. We don't know if that has anything to do with the disorder or not, but you can just imagine that that's the case when you don't want to be overly trusting of everyone.
I have a closely related question having to do with the vasopressin. I noticed that the quantity of it is important for pair bonding, but also it comes along with territoriality and aggression. And it reminded me of the very common cases where women will be involved in abusive relationships with men. And the question is, if the man is so angry at her so often, why doesn't he just let her go? And very often they're told that, he says he'll kill me if I leave him, or he'll track me down wherever I go, and so forth. And so it looks like if you get too much of that, you can have a very unhealthy pair bonding that involves a lot of aggression. And I don't know about marking territory, but certainly the territoriality.
I think men mark their territories sometimes.
Different ways.
But yeah, I think you're right. I mean, I don't want to really extrapolate too much to humans here. We can sort of think about that on our own. And, my expertise is in these animals. And I think that the formation of a bond from a male's perspective, the mechanisms in the male brain, may be a combination of this sort of territorial attachment. Okay, this is my territory, and I'm going to fight anybody who comes close, just like he would his nest or whatever. But there is also a component that may be this oxytocin dependent, which is more of a nurturing kind of component. And so those things can be in a balance in some way in the voles. So if the same sort of thing is going on in humans, you can imagine that you can have, in some cases, more of the sort of territorial possessiveness.
Yeah.
Or in other cases, more of the very nurturing kind of bond.
Yeah.
I have a practical question. Every year when I teach your material to my undergraduate students at NYU, an area that they're particularly interested in is the relationship between oxytocin and reward systems and opiate systems and addiction-like behaviors. And invariably, one student in my class stands up and tells me that she was recently dumped by her boyfriend and is completely distraught about it. And she always asks, is there an oxytocin-based treatment for lovesickness?
Is there an oxytocin basis for treatment of lovesickness? I don't know. I don't think that oxytocin is, because what oxytocin is doing is really allowing the linking of the social information with the reward. I think there are other processes that are involved in, like, social loss. So one experiment that we've done that I didn't talk about here today, but I think is really sort of relevant to bereavement and grieving. If we lose a partner, if we studied these animals after they formed this pair bond and then took away their partner. And then we studied their behavior, and they developed behaviors that are reminiscent of depression. So in animal models, we say it's depressive-like behavior. They did not struggle to get out of a difficult situation, for example. And we found that was related not to oxytocin, but molecules in the brain that are regulating stress responses. So CRF it regulates stress. So there are other systems that are involved in sort of loss of a partner. And you mentioned addiction. I happen to think that social bonding is very similar to addiction. It utilizes the very same kind of neural pathways. And I have 3 daughters who have been, various levels of teenager ages. And you can see in cocaine addicts, they have drug-seeking behavior, where everything they do is designed to get access to more of that drug. And I've seen my daughters engage in behaviors where all they want to do is to get access or talk to their boyfriends or friends or whatnot. But also in addiction, there's withdrawal. And there are physiological processes that happen in the brain following removal of that. And we see those same kind of things in pair bonding. So I think that's kind of a real common mechanism of 2 processes that have a common biological mechanism.
So Larry, you mentioned it was a wonderful talk, and one thing that's important, I think, for people who are being exposed to science and seeing these wonderful stories is that the results within a species aren't always the same, that we see there are differences within a species. So oxytocin, as you pointed out for these fake products, it's not a magic bullet, that we have a range of behaviors. So I was interested if you would comment on what else produces the range of behaviors within a species. So one vole will respond one way, and the next vole will not respond exactly the same way to the depletion or addition of oxytocin. So what other issues are at play here that give rise to the great diversity of behavior that we see as well?
So one that I briefly mentioned in my talk is early life experience. So the voles that were raised by a single mother versus mother and father, they have differences in the likelihood that they're going to form these social attachments. They have differences in how they display maternal behavior. In rats, there's been some really elegant studies done by Michael Meany's and group and many others that have shown that if you take little rat pups and the amount of licking and grooming that the pups get, so it's the amount of nurturing that the mother gives to the pups, has a big impact on how those pups will be mothers when they grow up. And that's partially through oxytocin system, but there's also a host of other neurochemical changes that occur during that development. So if you're asking, where does the diversity in behavior come from, or diversity in the way animals will respond to the system? Part of it is genetics, but the genetics do not determine behavior. They change the probability that an animal will behave in such a way. But part of it is also experience, which experience can act on those same genes that we've been talking about to change their expression to create this diversity in behavior. So, but neither one of those experience or genes predetermine behavior. They just change the probability that an animal will behave in such a way.
I have a question.
Yeah, Helen?
So you really made a strong argument about the difference between male and female behavior, and that the fact that you can switch the gene on the males and you can convert them from homebodies to hound dogs, hound bulls. And I was wondering, and that's happening in adult animals. What's the story on the gender diversity and changes in the genes for the moms? And can you actually make a homebody mom, not one that, you know, that was there to be the other way, to be adaptive, and when things to extrapolate to human behavior, that, you know, why do women adopt certain strategies and go back and make the same mistakes again? Is it just practical, environmental, or is it a little hardwired?
Yeah, I've done one experiment, or a couple of experiments that addresses that a little bit, and I'll just tell you about that. So I mentioned that some of the prairie vole females are spontaneously alloparental. They like pups already, and others don't. And they have the different levels of receptor expression. Well, using this viral vector gene transfer approach, where we put the oxytocin receptor gene into a virus, we can actually increase the expression of oxytocin receptors in the accumbens of females that have low levels. The first time we did it, we took adult females who had already, you know, they're already adults, and we just jacked up their oxytocin receptor expression. And we found that those animals, those females, would pair bond much more quickly now. So the machinery was in place that they would form an attachment better, but they did not show this increase in maternal nurturing behavior. So there's a dichotomy there. We then did a second experiment where we did the same injection, but we did it when they were 21 days old, so at weaning, so when they were very young. Then we found that when they grew up, they were spontaneously maternal, nurturing. So that suggests to us that there's an accumulative sort of effect of oxytocin stimulation. It's not just how much receptors you have now, but maybe the accumulation of the effects of those receptor stimulation over time has a more profound impact on sort of shaping the brain and impacting how you might respond in a social situation.
In the other direction, do you actually have experience where actually you lose your capacity, meaning thinking about the loss and some of the depressive behavior, that it goes in the opposite direction, kind of anti-prosocial, antisocial?
You mean, can we do a manipulation that makes them antisocial? We have not yet, but that's something that we're working on. Other genetic manipulations where we can turn down those receptors and see how does that change their behavior.
Any more questions from the panel? Okay, I'll go to the questions from the audience. Has anyone done any research looking at oxytocin's effectiveness in increasing empathy in people with sociopathic personality disorder?
I think that's a very good question, and as far as I know, no one has done those studies yet. But this comes up almost every time I talk, and so I think that this is something that needs to be done, to look at individuals who are sociopaths and see, well, might increasing oxytocin have any effect on them? But just keep in mind that in the studies where it increased empathy, it only did so for the say 45 minutes or hour and a half after the injection. So this is not a drug that's just going to, someone can take in the morning and increase their empathy. It may be that other drugs that can stimulate more chronically release may have an impact. But there's another question that I think is of interest is what about the receptors of individuals with the individuals that are sociopaths? Maybe they don't have receptors in the same locations as the rest of us, and so therefore they're not able to show that empathy. And this is something that we, right now we are not able to look into the human brain of a living person and see what the receptors look like, but we have been working for the last couple of years to develop PET ligands that can actually see those receptors, and we're making some good progress on that. So I think that in the future, that may be something that we can look at, not only in sociopaths, but if this turns out to be a mechanism that you can really target in autism and have a beneficial effect, you may want to visualize the receptors in children and individuals with autism to see do they have the receptors in the locations that may make them more likely to respond. So I think in the future that's going to be a cool way. A cool thing that we can do is to see what is your oxytocin receptor profile.
Thanks. Another one. You spoke exclusively of heterosexual pair bonding. What can be said about your research in terms of homosexual bonding?
That is a very interesting question that also comes up quite often. And Prairie Voles don't form homosexual bonds because the males are aggressive towards each other and they just don't do it. But I happen to think that the general mechanisms that are involved in this pair bonding process in voles, that they are somehow involved in human relationship formation, and that it probably is generalizable to all kinds of relationships. Even not only just, sexual relationships or romantic relationships, but I think it probably to some degree may be involved in friendships and, alliances and things like that. And there's been some studies to show that intranasal oxytocin affects something called in-group love, which is feeling a part of a cohesion of a group and doing things that would benefit the group even if it is hurtful to you. So I think it goes beyond just this pair bond. That's just happened to be what we study, but it's much more generalizable to all kinds of social relationships, I think.
It's time. We have one more time. How severe are the cases of autism that are experimented on with oxytocin, and is there a significant difference between the severity of the case and the effect of the oxytocin on their social behavior?
Right. So let me just say that we are only at the very, very beginning of exploring the possibility that oxytocin might be useful in enhancing social functioning in autism. There have been 3 or 4 published studies now that show some promising effect within that short amount of time after the drug is given, just enough to make us think that we need to explore it further. And in all of those studies, they were done in high-functioning autistic individuals. Right now, there are several other clinical trials that are going on to explore in more detail, how we might use this. So I would imagine that the higher functioning, the better, and the earlier, the better. But one point I do want to make is people should not be taking this science now and using it as a rationale for maybe giving their child oxytocin, or even psychiatrists. I've heard that psychotherapists around the country are thinking about using this, or in some cases actually giving it to children. That absolutely should not be done until the science is all in and we know under what context it actually works, and to make sure there are no detrimental effects of long-term use. Because all of the studies that I showed you today in my slide with the 3 different groups, they were all after a single administration, and then examining how that child functioned within 1 hour or so afterwards. And so based on that, I do not think it's a panacea to just increase social functioning and autism in an autistic child. But it may be potentially useful in the right way in the context of these behavioral therapies in a very controlled situation. So, we're just at the very beginning of this, and I think we're going to learn in the next few years, what is the optimal use, if it is useful at all.
I really look forward to that. I want to thank the audience for their questions and Dr. Young's fellow panelists for their questions, and I would like to have everyone thank Dr. Young for answering those questions. Thank you. I think music will be resuming at 12:45, and our next speaker, Dr. Ramachandran, will be arriving via teleconference at 1 o'clock. Thank you.
$20. You have